So, welcome to the last session of this workshop on models old and new. And we have glad to have with us Ira Longini from Florida, who is going to talk about the vaccine development. The entire title is there on the screen for you. So Ira, you have about half an hour for your presentation, and begin as you like. No, welcome.
Yeah, so thank you, Dennis. It's a pleasure to be virtually back here at the Newton Institute. And I think before I get started, I'd like to note that I gave a talk, the first talk at the Newton Institute in 1993 when we had this inaugural meeting of infectious disease modeling. I gave that talk on HIV vaccine trials. And I was very optimistic about going into phase 3 and having an HIV vaccine. And that was 27 years ago, and we still don't have a HIV vaccine after many failed trials. So let's hope things are better for the COVID vaccine, and I don't jinx it by yet giving another Newton talk on this topic. So today's, I'm going to talk about a large international multi-center adaptive randomized vaccine trial for COVID-19, some dynamic statistical details for finding one or more safe vaccines at warp speed, which is, faster than even Donald Trump says we'll have a vaccine. So first I thought I'd define warp speed to get started. So there it is. We're moving at warp speed now, which is about 300 kilometers per second. And so warp speed is roughly 9 times 10 to the 10th kilometers per second. But what does this mean for a vaccine? It means we'd like to have at least one safe and efficacious vaccine 3 to 6 months after beginning of a randomized efficacy trial. So that's warp speed. Here I've listed more than 100 vaccines are under development. The count here is 159, and I'm showing the different. Can you see my arrow? I'm never quite sure when I share the screen.
Yes.
Good. So these are the different platforms going from non-replicating viral vector through DNA and RNA vaccines, inactivated vaccines, and so on. Other ones, live attenuated vaccines, and you can see some others too. And they're all in different stages of development. But the two that are the closest now to phase 3 vaccine trials are, there's a vaccine made by Moderna. It's a US company. It's the RNA vaccine. And this is the one referred to as hopefully going in warp speed. It's a 2-dose vaccine. It's in phase 1 currently. And I want to note that there's never been a successful RNA or DNA vaccine developed for humans after many years of trying. The second one is this Oxford chimp adenovirus vaccine being developed at Oxford University with partners. And it's a replication deficient chimp adenovirus with a SARS-CoV-2 spike protein. It's a 1-dose vaccine, and it's already in a relatively large phase 2 trials, and should probably be the first out of the box, probably. Before I go into these vaccines and the vaccine trial, I want to talk a little bit about modeling using some joint work with Alex Vispignani. I don't know if he talked about this model or not when he gave his talk. But it's really about at least what can we do with no vaccine or effective therapeutics, which is the current reality. And so this mathematical model, it's a large-scale agent-based model set up with layers of social contact and distancing and so on. What I'll show you is some results using this model. It's a the usual SEIR model for the Boston area, and it looks at different strategies for mitigation while we wait for a vaccine and therapeutics, and just kind of showing the realities of what we can expect in much of the Western world that's already experienced a large first wave of the epidemic, including Sweden, I should mention. And on the left shows the epidemic as simulated in, and this is Boston, but you can pretty much apply it to anywhere in in these populations in the West that have had large epidemics. But on the left, you can see, as we lift essential, partially lifting the essential distancing, first putting it in place, you can see the curve coming down, and then the partial lifting. And as soon as that happens, if we simply lifted, we get this epidemic, this dashed line, which is a large second wave. Here showing the epidemic curve, and over here the cumulative incidence, and over here showing the effective reproductive number going above 1. And then the solid line shows what would happen if starting in mid-May, if we did 50%, we were able to do, through testing, find 50% of the symptomatic cases, and then quarantine a proportion of their contacts. And the solid line shows what would happen if we could get 20% of those contacts. And the dotted line down here shows if we could get 40% of those contacts. So in both cases, we could actually contain the epidemic fairly well and just stretch it out. And again, this over here showing the effective reproductive number as we do that. So that's the best we can hope for in places like the US or Great Britain or even Sweden. And so hopefully that's going to be what happens as these different populations lift their restrictions. But now back to the vaccine. I'm going to describe to you the work on getting this trial designed and into the field. And this is a joint work with the World Health Organization, the so-called Blueprint. And we published the paper, the group of us working on this, in the New England Journal of Medicine earlier this year, showing basically the framework that we would need for conducting randomized clinical trials, vaccine trials, in the face of an outbreak. Just to familiarize you quickly, if you don't remember what the phases of a vaccine trial is, phase 1 is a small scale, usually the tens or up to 100 individuals, trial for just safety and immunogenicity. Then phase 2 just extends phase 1, and that is for larger scale safety and immunogenicity, usually dose ranging studies. And then given that those are successful for a certain product, then it goes into phase 3, which is large scale efficacy trials, where you actually find out if the vaccine works. The idea of accelerating these COVID-19 trials is to abbreviate phase 2 or even do away with it. The background for doing this is the WHO R&D Blueprint to Prevent Epidemics. And the blueprint, which I've been working in for the last 5 years, has prioritized various important emerging infectious diseases. So this was done several years ago. And that's a list of the diseases that we've been working on to try to develop vaccines and therapeutics for, and test those in the face of transmission. And you can see that in the middle that these different coronaviruses we were already working on those, both MERS and SARS-1. And then down here you can see disease X has always been here, which is an unknown pathogen, which is just emerging, but we don't know what it's going to be. And so, as I mentioned, given all this, at WHO, we've worked on this idea of, we call it a solidarity trial for accelerating the safe and effective COVID-19 vaccines. And there's also one of these for therapeutics, which is in the field now, and also several for antiviral agents to be used as prophylactic, both pre-exposure and post-exposure prophylaxis. So what will this novel coronavirus vaccine trial look like? That's going to be a large international randomized controlled trial with multiple vaccines adaptively evaluated, and adaptively is an important term as we plan to evaluate more than one product, more than one vaccine at a time. Different vaccines will be available or suitable to enter the trial at different times. And for each vaccine, the primary efficacy results are expected within 3 to 6 months of the vaccine entering the trial. The precedent for doing this goes back to the Ebola epidemic of 2014-15 in West Africa, where a VSV vaccine, vesicular stomatitis vaccine, was developed in a vaccine trial in Guinea, a ring vaccine trial in record time. And this shows the epidemic curve and the timeline. We started, the idea was to get a vaccine trial into the field late in 2014. The trial actually started in April 11th of 2015. We had an answer by July 2nd, 2015. So that was 2.5 months for the phase 3 trial, which is right here, which caught the descending curve of the epidemic. It was a ring vaccine trial, which means vaccine was allocated to rings. Those are individuals in contact with a primary case, and individuals rings either got immediate vaccination or delayed. And then the efficacy was estimated by comparing the rate of illness in the immediate rings to the rate of illness in individuals in delayed rings before they got their vaccine, which was 21 days after entering. So what was a ring? There was an index case. There were close contacts. There were people who lived in the same household. People visited the symptomatic patient, extended family, household members of high-risk contacts, and others, neighbors. And so it's exactly the kind of tracing we do for COVID-19 as well. So this is a definite design that could be used for COVID-19 vaccine trials. This just shows the result at the interim analysis, the Kaplan-Meier, or 1 minus the Kaplan-Meier cumulative incidence, where the dotted line shows the vaccinated arm, vaccinated arm, and this curve up here shows what happened in the unvaccinated. And so the estimate of efficacy, which was the primary outcome for this vaccine, was 100% with this confidence interval and that p-value. That p-value is almost sufficient for stopping the trial at the interim point, but the Data Safety and Monitoring Board decided to stop anyway. As I said, 2.5 months after the trial went into the field. And this was a secondary outcome, which was measuring the overall effectiveness or overall reduction in rings that got vaccine compared to those who got delayed. So this includes both vaccinated and unvaccinated people, and this efficacy was 75%, but the trial was not powered to detect this as a secondary outcome. This was published in The Lancet on July 31st, 2015, The vaccine since then has been licensed. It was used to control the epidemic in the Democratic Republic of Congo, the Ebola epidemic that we just had. Okay, so for the current trial, this is what it's going to look like. So I'm going to show you the statistical details and some of the dynamic details of this design and what's to be expected. So the primary outcome will be laboratory confirmed COVID disease. The trial is set up to detect vaccines that we want to reject vaccines that have a 30% or lower efficacy against those that have higher than 30% efficacy. So the null is 0.3 and the alternative is greater than that. And the efficacy will be estimated as 1 minus the hazard ratio comparing the vaccinated to the unvaccinated individuals. The secondary endpoints will be, there's quite a few secondary and exploratory endpoints. I should note that all the participating sites will, of course, do the primary endpoint, and then they will also do efficacy against severe disease or death, efficacy against disease for subgroups, especially the elderly, and long-term efficacy against waning disease and enhancement, with any alpha that's left over at that point should a vaccine be successful. And we're very concerned about enhancement for these coronavirus vaccines. It's quite possible, but hopefully it won't happen, that vaccinated people, as their immunity wanes, could be set up for more severe disease, and sorry for misspelling disease here, than those who are unvaccinated. Then there's a whole host of exploratory endpoints that all the sites will not do. Necessarily will be infection, shedding, immune correlates of risk, and surrogates of protection. Randomization for this will be double-blinded in the sense shown here. So it's a rather interesting and unusual setup since multiple vaccines will be tested at one site. So it's going to be done such that people can be vaccinated. They'll be randomized to a vaccine, and these different vaccines will very well have different placebos. So they'll be randomized to a vaccine placebo combination. So they get the placebo for the vaccine they're randomized to, and then so they'll either, once they're randomized to the group, then they'll either get vaccine or placebo, their particular placebo, but then the placebos will be pooled for, and everybody who's assessed for efficacy will be assessed against the pooled placebo. So this just shows some time windows quickly. Suppose the vaccine is, one vaccine's available, then that person will either get the vaccine or placebo, and the matching will be 1-to-1, both for the matched placebo and the shared placebo. Suppose a second vaccine comes in, so there's two. So this shows all the combinations of which individual could get vaccine A, vaccine B, placebo A, or placebo B. So the individual matching will be 2-to-1, vaccine to placebo. But again, the individual shared comparisons will be 1-to-1. And this will be the case when 3 vaccines are present. And we just keep cascading up, vaccinating pretty much, if there's K vaccines, then there'll be K+1 randomizations. This shows the sample size, how the trial will be sized. For example, if the efficacy had we're designing it for an alternative of 60% efficacy or higher, it would take 150 total events for every pair of vaccine and placebo to get to the N with a 0.2025 one-sided p-value. And this shows different illness attack rates in the first 3 months. If we could get a 1% cumulative attack rate, illness attack rate, then we would need about 40,000 people, 20,000 in the vaccine arm roughly, and 20,000 in the placebo arm. And we would need this for every pair of individuals. So if there's 2 vaccines, then we would need 20,000 in each vaccine arm and 20,000 in the paired placebo. Okay, so this just shows sample size for different cumulative incidence or attack rates. Trial could be as big as 172,000 if the attack rate was quite low. And then this is for a higher alternative efficacy, but the red is what we're powering the trial for. So roughly 150 events and roughly 20,000 individuals per vaccine arm. Group sequential rules will be used for the interim analysis of this trial. So we're planning for 2 interim analyses for success or futility. And so 1/3 of the way through will be the first interim analysis, 2/3 of the way through will be the second, and then there'll be a final analysis. And we're using one-sided O'Brien-Fleming boundaries for alpha spending. And so, for example, and I can't show you these boundaries because they'll because they're secret at this point, but you could probably work it out if you had some time. But the basic setup would be that at 1/3 of the way through, or 50 events, all the vaccines that hit 50 events, when they hit 50 events, would be assessed for efficacy and then for futility. The vaccines that fail due to futility would be kept in the trial, but we will no longer randomize to that vaccine or its placebo. Then 2/3 of the way through at 100 events, and again, this is event driven, so it's different when each vaccine hits 100 events total. That's both in that vaccine and the shared placebo. Same thing will be done. And then finally, at the end, when each of these hit 150 events, then you can see the boundaries. I've written them here because they're basically what I've already stated, that the goal will be to find all the vaccines that have an efficacy of greater than 50% and eliminate those who have efficacy less than 30%. So we would hope to weed out those with the lower efficacy, less than 30%, and find those that have an efficacy of at least 50% or more. Then the DSMB would stop randomization at that point, but we would continue to follow these individuals, including the placebo arm, until one of the successful vaccines is actually available in the region where the trial participants are located. So that's the basic trial setup, and the dynamic, adaptive design, and how things will be orchestrated as we go through this trial. How will we select the vaccines? WHO has a special process for doing that. So vaccines are going to be scored and ranked using the following criteria. Safety, no vaccine, of course, will get into the trial unless it's shown to be safe through animal studies and phase 1 data, and phase 2 if there's any phase 2. The vaccines will be based on their immunity profile, and animal challenge studies and other criteria will be assessed for whether they're thought that they will be eventually efficacious or not. Vaccine stability will be looked at, especially the temperatures in the cold chain. How implementable are these vaccines? Do they need special implementation equipment? Some of the DNA vaccines require extra implementation. And then quite importantly, no vaccine will get into the trial unless they've shown the ability to make large quantities of the vaccine and distribute the vaccine if it should be shown to be successful. The other important thing that needs to be done, and there's yet another group at WHO doing this, will be to actually screen for trial sites to be selected for the trial. And it's going to be a combination of fixed sites, which are sites picked ahead of time, and pop-up sites where mobile teams will go and vaccinate should outbreaks occur in these pop-up sites, including ring vaccination. And so the criteria for selecting sites is that sites need to be able to show that they can maintain a 1% or higher COVID-19 illness attack rate over 6 months. And how will we do that? That's going to be a combination of surveillance for the virus, serosurveys to assess how much immunity is already in the population, and modeling projections. And we're planning to use ensembles of models that project transmission in different parts of the world, models constructed by different modelers, and try to use these models to predict which areas are going to experience high attack rates in the next 3 to 9 months. Of course, as I said, so site selection depends heavily on that potential for future outbreaks, and sites have to demonstrate the infrastructure to support a large vaccine trial. So we're talking probably about a trial in hundreds of sites in dozens of countries involving hundreds of thousands of people, maybe up to as many as 1 million people before it's over. To quote the Beatles, there will be an answer. Hopefully this trial will start as early as July 2020, and we'll have an answer as early as October 2020. There's no reason why this can't happen. We'll have all the infrastructure in place, and I strongly believe we'll have phase 3 trials in the field by then. Of course, this is all done voluntarily by the different pharmaceuticals, but we're hoping a good part of the world's pharmaceuticals that have these vaccines will join this solidarity trial. So thank you.
So I was there in 1993 when you were making those projections about HIV. I've never forgotten the lesson of that. And so it seems like your piece looks like it's extraordinary well developed. So I have 2 questions. One, is there anything behind the curtain on the things that you've presented just this morning that could potentially derail the strategy that you've identified? So you tend to be very optimistic about this stuff. I know you sort of over the years. And I think then the second question is, in terms of derailing, all of this relies on having something that's ready for a phase 3 trial. So what do you see as the main obstacles to getting any of these candidate vaccines to that? I noticed, for example, the 2 that you identified as being sort of closest to that boundary right now. One of them, the RNA vaccine, we don't have a single RNA vaccine, right? So what are the, if we step back from the optimism here, what do you see as the biggest threats in both the getting the vaccine to phase 3, and then if we are able to start phase 3, where's the biggest challenge going to be?
Yeah, so I don't see any big threats. Well, one big threat is Donald Trump, because
yeah. Right.
And is he in the U.S. and will the RNA vaccine even participate in this? since the U.S. seems, determined to do its own trial. Again, I don't know where the trial would be done, and how it could be pulled off. So one threat definitely is this
Politics.
lack of international cooperation that we're seeing mostly from the U.S. And that's quite a serious threat. Other than that, I don't see, as I said, these products, this is all voluntary, pharmaceuticals. In the end, the decision is up to the pharmaceuticals, not anybody else, where their product's going to be tested and how it's going to be tested. And we've done everything possible to make it clear to the manufacturers that their vaccine will be fairly evaluated. There's no head-to-head comparison. And the shared placebo makes it extremely advantageous to participate. So I don't see an operational threat other than the lack of organization internationally perpetuated by one particular government. But as far as these products, yeah, I did express some pessimism about RNA and DNA vaccines, and I think that's well founded. It's probably quite likely that product will not be in this trial. It'll be in some kind of separate U.S. NIH trial, which is fine. But then, of course, you lose this big advantage of, because not only do you have in the Solidarity trial everything under one umbrella, but you have a single data safety and monitoring committee. You can have much better evaluation and much more balanced of a product in that situation, and having many separate trials, as you know, tends to lead to chaos. So I don't see any strong threat. I think the enhancement issue is a big potential showstopper for all these vaccines, and we just have to evaluate that.
Yeah, just one comment on that. If there is enhancement, and correct me if I'm wrong, but it seemed like enhancement was fairly far down the list of the secondary or tertiary outcomes that would be evaluated in every site. It's exactly the kind of thing that the anti-vaxxers would jump on immediately. So it is, I think it's a threat in many different ways.
Yeah, it's a secondary outcome. And one problem is none of these trials will really be powered to assess enhancement, which could come quite, a year after vaccination or even a couple of years. And it's still, if it occurs, it's still a relatively rare event. You just start to see an excess, a small excess generally in severe cases in vaccinated compared to unvaccinated people. And our last experience with enhancement was with the dengue vaccine. And again, it was quite a rare event. So although enhancement could be serious in terms of the outcome, it may not be serious in terms of numbers. So we cannot power these trials for enhancement as a primary outcome.
Carl, you had a question.
Yeah, hi Ira. Thanks for the talk on the trial stuff. So I realize this isn't exactly a science question, but maybe you can make it into one. How would the approach that you've discussed potentially dovetail with the approach that some others have suggested of doing a human challenge trial?
Yeah, well, I think this would be complementary with human challenge trials. You can do a human challenge trial, and I don't know.
Can you explain what a human challenge trial is? Not everybody knows.
Yeah. So you vaccinate. You can find volunteers. You vaccinate them with vaccine or placebo, and then you give them a direct challenge with the virus. And you really have to, that's where the rub comes, because what virus? Is it a fully virulent virus for which we have no known therapy? Or is it a laboratory, attenuated virus? How big should the dose be? All these things. does it look anything like the dose one would get out in the world? So even though human challenge studies can be good for proof of concept, in the end, you still have to do efficacy trials to find out if the vaccine actually works, given the viruses that are out there and the types of challenges people have in the real world. So I think human challenge studies, just like animal challenge studies and primate challenge studies, are good proof of concepts. But of course, they're better than animal challenge studies, but they still don't prove that the vaccine will work when used in under real circumstances. Of course, and if they fail, they don't prove it doesn't work, because usually the challenge dose and strain may be quite different and much stronger. You know, you're giving somebody a direct dose of the virus rather than them picking it up from the environment.
So you sort of see it more as if those kind of trials, if people are able to make them happen, they still become just additional candidates in this multi-arm. Right. Okay.
And you know, regulatory agencies do not allow animal challenge studies or immunogenicity data or anything like that to substitute for a phase 3 trial. And so in the end, you have to do a phase 3 trial if you can do one. If you can't do one, then there are other pathways to licensure and approval. But if you can do an efficacy trial, you have to do that.
Peter.
Yeah, thank you for the nice talk, Ira. So I had a quick questions, playing the devil's advocate a bit, but in the, was it 2011 thing, there came from Finland and Sweden some concern that there was a relation between necrolepsy and the vaccine, one of the vaccines tried, and my common sense says that no vaccine, you say a vaccine should be safe, but it does do something to your immune system, so nothing is 100% safe. There is always something which can go wrong. So how can you reasonably exclude risks of like necrolepsy, or if you bring a vaccine on the market within months?
Well, first of all, the phase 3 trial, which will be in, as I mentioned, hundreds of thousands of people, safety is an important secondary endpoint. In fact, more than a secondary endpoint, so safety will be monitored very carefully in these trials. And I could show you, we're very well powered to even pick up rare events with this large number of people.
But it is rare events which happen within the 3 months, right?
Right. So that's always a concern with vaccines. And of course, there'll be phase 4 large population studies of these vaccines that are successful as they're deployed.
Thank you.
Hi, Ira, thank you very, very much indeed. I'm not a specialist in this particular area, but I do a lot of different statistical public policy things in my life. Did I understand you to say that the trial has to, you can only really try out a vaccine if you've got a reasonable percentage of infection in the population? I mean, it's obvious anyway. And I read somewhere that that's one of the problems that the Oxford trial may be having. By the way, the background you can see is Oxford, which is where I'm sitting, but I have nothing to do with it. Of course, if all these lockdown or whatever strategies work, then there won't be anywhere which has 1%. And so the human challenge may, as it was in the case of things like smallpox, be absolutely necessary. My hunch is that there would be no shortage of volunteers, actually, given that we know that the vaccine, given we know that there'll be young people. And of course, then the question arises, what if the vaccine's only effective in young people? And then you can model all that and so on. So the question is, do you actually think we'll get away without doing human challenge? And then the second, which is completely non-scientific question, but just curious, if a state governor allows a state to take part in a trial, presumably the federal government can't stop them. But that's another question.
Yeah, so with respect to enough incidents, that there are really large epidemics, many of which haven't even reached their peaks in Latin America, in India, Southern Asia, Africa. So I don't think we'll be able to do these trials effectively in the parts of the Western world, developed Western world that are locked down, and it would probably take forever, which is fortunate. The unfortunate part is, as they loosen the lockdown, the epidemics will get bigger, and then we don't know what will happen in the fall. But I don't think there'll be a shortage of populations to do this, and if it starts in July, for example, and we can get an answer in 3 to 6 months. Obviously, if we had to only go with human challenge studies, I don't know. It's a big gamble to make billions of doses of vaccine based on evidence from human challenge studies. But if we had to, we have to, and we'll probably do something like that. And then I don't think in particular regions of countries, it's really a local decision whether to participate in these trials from the WHO point of view. It's a regional, local decision. So I don't think, these locations would need permission from the national government.
Yeah, I mean, good luck. And thank you very, very much indeed for a very interesting talk. And the sooner we get on, in fact, I'm wondering whether at the age of 68 in good health, I would volunteer for a human challenge study. Yeah. And other people in the Zoom chat will say, Bernard, don't even think of doing such a thing. Mm-hmm. So thank you.
Yeah. Thank you. Thank you. I would mention, I think someone else also brought it up, that these trials will be big enough that we will certainly get efficacy data in the elderly. Yeah. Although we're not going to target the elderly for these trials.
Don't you dare call me elderly, I'll log off immediately. Thank you.
Martina.
Yeah. Oh, I think Claudio's had his hand up.
Oh, sorry, Claudio first. Okay, yeah. Then Martina.
Ladies first. Martina, go ahead, and then I go after you.
I've already asked a question. You go, Claudio.
Okay, so hi, Anna. So if you are still waiting for an HIV vaccine, I am still waiting for a malaria vaccine, okay? So we have 10 more years than you have been waiting an HIV vaccine. So I would like to go back to the ADE discussion, the antibody-dependent enhancement that Martina brought up. So the idea is, I would like to draw an example from the dengue vaccine. So in the dengue vaccine, we had the first trial, and the face values of this, the main estimates of those trials were showing that the vaccine was marginal at best, and it was bad among children. Okay, then came a second wave of work with modeling the strategies to use the vaccine, and the main outcome of this exercise was to recommend vaccination. And when vaccination actually started, we saw that this was not a good idea. So I wonder if ADE is an important component in that, even if it is rare. If we would be following the same example of the dengue vaccine, and how can we prevent this kind of pathway?
Thank you, Claudio. And I've been a part of that dengue story from the beginning as well. Went through all that. I think the ADE mechanism for the theoretical one for coronaviruses is different than for dengue, you know, being an acute infectious disease. And so I think we probably see it faster if it's going to occur. So we should be able to pick it up in these phase 3 trials, even though they're not necessarily you know, powered directly for ADE. But it is a theoretical concern, and we're just going to have to deal with it. And if we do get a vaccine, it shows up later, you know, then we'll get into this whole cost-benefit weighing exercise that we did with the dengue vaccine. Which, by the way, I'm not convinced that vaccine would probably, if widely used, would probably do more good than harm, in my opinion. But that's just an opinion. And then, of course, we could use it by testing people first. So I just think it's a complicated issue that, you know, we'll just have to deal with as, you know, data come in. You know.
Thank you. Are there any more questions? If there are no more.
Oh, Martina has one.
So I find it interesting that the idea is to potentially eliminate the phase 2 part of the trial sequence, which is safety and dosing, right? If I'm not mistaken. Do you think this is only when we need to go warp speed, or does this suggest a more general approach to shortening the pathway by evaluating safety rigorously at steps 1 and 3 as well, so you don't need a separate step?
Yeah, it's clearly part of the warp speed. We need a vaccine quickly for this virus, and so, and also maybe it could be a new paradigm if the phase 3s were huge, like these may be. So we could pick up a signal maybe in the first 100 or so individuals vaccinated if there is a safety signal. And originally the protocol did have that built in, that we wouldn't assess efficacy in the first 100 or so vaccinated people. But we dropped that because it just became too complex to, you know, compare all these vaccines with this burn-in period for safety. But yeah, it has to do with speed. We're willing to sacrifice a bit for safety for the speed.
Any more questions? If there are not more questions, we should thank Ira very much. And I understand you have a busy, well, it's a morning where you are, but we hope you'll engage with the program during the coming months. So thanks very much for coming today and speaking to us. Thanks, Ira.
Okay, thank you, everybody. It was a real pleasure, and great to see you all again, many of you.